All right, let's discuss that cytoskeleton. So the cytoskeleton is made out of three different kinds of fibers, three different kinds of tubes, I guess you could call them. The largest of which are the microtubules, the smallest are the microfilaments, and then the ones in the middle are called intermediate filaments, as the name implies. We're going to discuss the microtubules first. Let's check them out. All right, so microtubules, super important. Made out of tubulin. The protein they're made out of is tubulin. You don't need to worry about the diameter. You don't need to worry about the 13 columns or anything like that. What I'd like you to know is that they're hollow. All right, so that actually does matter, that they're hollow. And let's go to this here. All right, and the fact that they're hollow cylinders makes them really strong. Because of that, they have this ability to prevent the cell from getting distorted. They resist bending, essentially, is what they're all about. So that's kind of the low-tech, simple function, a structural function that they have. But they also have quite a few really important functions like motility, mitosis, and movements of organelles inside of the cell, all of which we'll address here. So first one we're going to talk about is the idea that spindle fibers and centrioles are made out of microtubules, and both of them, when you look at them in cross-section, have kind of a star shape, kind of a pattern to them. Centrioles have essentially three microtubules in each ray, and there's 9 total in the centriole, and they're kind of twisted. Like if you look at the star itself, it looks like it's kind of twisted. Kind of actually a beautiful shape. And when you look at these with an electron microscope, they actually look exactly the way we expect. They have this kind of a twisted star shape of 3 bundles of microtubules in each ray like this, arranged. All right, the function of centrioles, we'll talk about at a different time when we talk about mitosis, but it's actually kind of mysterious. We don't fully understand what they do. They have something to do with cell division, though. All right, the other structure that has these microtubules in a very similar pattern is the flagellum, and along with that, cilia. Basically, cilia are just short flagella. These are tail-like or little hair-like structures made out of microtubules that help cells swim or move through the water, or they actually move water past the surface of the cell. Depends on the situation or depends on the cell type that they're in. So here we have 9 sets again, but this time it's doublets. So we have not triplets of tubules like we did with a centriole. What we have instead is doublets arranged in this, once again, star-like, twisted star-like kind of a pattern. We also have 2 in the middle, 2 single ones in the middle like this, so we call that a 9+2 configuration. It's really 9 doublets, so it's really technically 18 on the outside, and then the 2 in the middle, but they call it 9+2 configuration. And the 2 in the middle, they kind of add more strength or structural support. When you look at this with an electron microscope, they look just like what you'd expect. So this is an actual electron micrograph right here, and it looks basically the way it would look in a textbook with a drawing made by an artist. All right, so the flagellum then is anchored in the cell membrane by this thing called a basal body, and the basal body has the same configuration as a centriole does. It has the 3, sort of this triplet with 3 of them like this, and then there's 9 that make up the good old star, twisted star kind of shape again. So basically centrioles and basal bodies, the way these things are anchored, the flagella or cilia are anchored in the membrane, they have exactly the same structure. And that's not a coincidence, because the first centriole in a cell, or in an organism, I should say, organism cells, is the basal body from the sperm, which fertilizes the egg. As that sperm penetrates in here, it donates the DNA, and so the chromosomes, and it donates its basal body as the first centriole. So the structure of that, the similarity is not a coincidence. It makes sense. All right, let's talk about flagella in a little bit more detail, and how exactly do they move, how do they whip back and forth. And the way that works is that each doublet of microtubules, each of these rods here, is connected to the next one with special motor proteins. And these motor proteins are called dynein. Motor proteins that just means that they are able to move or cause movement. So what happens is at the expenditure of ATP, these motor proteins will grab on to the neighboring doublet and then push it in a certain direction. So they essentially push it past the other one. But there's also these connecting protein called cross-linking proteins that prevent this just from shooting out of here like a dart from a crossbow. What happens is they're sort of trapped, and then instead of just shooting out of here like this really fast like an arrow, the whole thing bends. So there's pushing in this direction, and then there's also this connection that holds them together, causing the whole thing to bend. So first it bends in this direction, and then the process is reversed and it goes the other direction. So they go to the right, then they go to the left. That means the flagellum just keeps sort of undulating back and forth, and it's able to propel the cell through the water and cause movement that way. All right, so this costs ATP. Each time the little dynein molecule ratchets along the other microtubule, each time it makes one of these movements to the new position, that costs 1 ATP. So you can imagine how many ATPs are used when thousands or millions of these are doing this at the same time. Cilia work exactly the same way. They're just shorter. Anyway, interestingly, eukaryotic cilia and flagella have a membrane around the outside. So the membrane, sort of goes all. So if this is the cell right here, and if there's a flagellum right here, the membrane covers the entire flagellum like this, and then covers the rest of the cell. In bacteria, there is no membrane around their flagella, so structurally they're a little bit different. All right, another example of microtubule function is movement of organelles or vesicles inside of the cell from one place to another. So we know that vesicles get made by the rough ER, and we know that they get modified and processed and packaged by the Golgi, and those vesicles then have to move to other places in the cell, like to the membrane. The way that happens is that microtubules guide them along their path like railroad tracks, and there's a tiny little motor protein, kind of similar to the dynein. This motor protein is called kinesin, and that motor protein sort of walks along the microtubule and carries that vesicle with it. It's attached to the vesicle and it drags the vesicle in this direction then. Each step of the motor protein costs 1 ATP. So let's take a look at that. Apparently I have to go back to arrow. All right, so now I can select this, and let's see if we got a little video here. All right, so what we got here is our little, here's our little guy, our little kinesin motor protein. He's right here, and he's dragging this gigantic vesicle along the microtubule, and this is the microtubule right here. Each step that he takes costs 1 ATP. So while this is a cartoon, it actually is somewhat realistic. Maybe not with the colors, those are artificial, but the shape of the structures and the shape of the molecules is realistic. That's based on molecular modeling, and they used a computer then to sort of animate the process here, but this is what they would look like in terms of their dimensions and shape. All right, there goes our kinesin. All right, here we go. So that's another function of microtubules is intracellular movement of materials, or specifically vesicles. And I skipped the mitosis part, so let me add that in here. Let me add a slide to add the mitosis. Here, we can put that in the title. All right. And I'll just draw this for this part. All right, so we all learned about mitosis freshman year, and we know that at metaphase of mitosis, I'm just going to draw at metaphase. I'm going to skip all the rest. All those chromosomes are lined up in the middle of the cell. We got our centrioles over here doing whatever it is that centrioles do. There's two of them. There's a pair of them at each side. And then there's these lines that connect, these sort of ropes or cables that connect to the sister chromatids like this. They seem to be going over here to the ends or to the poles of the cell. Okay, so those things are called spindle fibers. And the spindle fibers are nothing more or less than microtubules. So one spindle fiber is one microtubule. It's just that in this application we call them spindle fibers. So there's three kind of spindle fibers you're going to see. There's kinetochore. Kinetochore fibers directly link to the chromatids and then pull them. All right, so then we also have polar fibers that go all the way across. And they sort of push the cell apart and elongate it. And then we have these aster fibers that kind of anchor the centrosome over here, along with the centrioles, to the membrane. So those are called aster, which is kind of short for asterix, which means star, because they kind of look like a star. All right, so that's another application of microtubules. Sorry, I didn't have a slide ready for that, but yeah, whip one out. So mitosis and pulling the chromosomes, spindle fibers. All right, let's move on. The next and smallest of all the fibers of the cytoskeleton would be the microfilaments. Microfilaments are made out of actin. And actin is a really common and really famous protein. It's the protein that's in muscles and helps us to contract our muscles, helps us move. But actin is much more universal than that. It's basically most cells that exist have some kind of actin in them because it has a bunch of other functions, not just muscle contraction. Look at all these functions. So one primitive, simple, low-tech function, once again, is maintenance of cell shape, but actin is elastic. Unlike microtubules, microfilaments are elastic and they can bend and then spring back like a spring or like rubber, like a rubber band maybe. So if a cell does get distorted, the actin allows it to then return back to the original shape, kind of like memory foam. Actin is a double helix made out of these actin filaments, but that's not super important. The size is not important. Don't worry about that. I'd like you to know that it's actin. That'd be nice. And I'd like you to know these. So let's talk about these in a little bit more detail. All right. The most famous and easy to understand one is probably muscle contraction. So there's two proteins involved. There's myosin and there's actin. Those two work together. The fat fibers, these guys right here, are myosin, the purple ones. And then these orange skinny ones are actin. Myosin has these cross bridges that kind of act like motor proteins. They're kind of like the kinesin and like the dynein. They reach across to the actin and they push. They pull or push on the actin by ratcheting back and forth, and each time it ratchet costs 1 ATP. Each one takes 1 ATP each time it clicks across. And then it basically pulls this orange one this way and it pulls this orange one this way, causing the entire muscle to get shorter. All right, so there's lots of these in each muscle cell, and then there's lots of muscle cells in a muscle that all sort of work together to make this happen. Interestingly, muscles can only shorten, they cannot extend on their own, so the muscles have to get pulled apart again passively by another muscle, by the antagonist muscle. So for example, in the arm, in the human arm, we have the biceps, okay, which causes the hand to go this way and for you to bend your arm. Then we have the triceps down here, which then causes you to move the arm back and straighten it out, which then extends the biceps. That's the worst drawing I've ever made. Let's ignore that. All right. All right, some more functions of actin microfilaments. One of them is amoeboid movement. So amoeboid movement is the movement of certain, not all cells can do this, but certain cells can do this, like amoeba and some related cells, and like white blood cells, where what happens is the cell shape literally will change in order for the cell to move. So it extends these pseudopodia, which is false feet. Pseudo is false, podia is feet. It extends those in the direction that it wants to move. And the way it does that is all these actin filaments right here, all these microfilaments, will sort of cross-link together and pull the membrane this way. And if they pull the membrane this way, it squeezes the liquid in here in this direction, kind of like you're squeezing a water balloon that bulges out on one side. All right, so the outer sort of realm right here, close to the membrane, can turn into what's called a gel. It turns real thick with a high viscosity. And then the inner part, by the actin filament sort of relaxing and letting go, turns into a sol, which is a fluid with low viscosity, which then allows it to easily be squeezed in this direction and allows these pseudopodia to form. So actin filaments basically pulling on the cell membrane, causing it to change shape, allow these kinds of cells to do this amoeboid movement. And yes, we have a little short video here. They do not make it easy for me to switch away back to the pointer. Okay, here we go. So this is an actual amoeba. Amoeba proteus is the species, and you can see pseudopodia forming. There's two of them forming right here at the same time, sort of towards the left here. You can see little tiny organelles and particles, vesicles, and things like that in there. There's some more pseudopodia forming. You see those streaming around in there. That's all thanks to that actin on the right side squeezing and then pushing it in this direction to cause it to flow, essentially, or creep to the left. All right, that's good enough. All right, another function that's kind of related to that is, it's very, very similar, to be honest, is cytoplasmic streaming. And this happens in some cells. And a good example would be plant cells, where the organelles inside of the plant cell and the chloroplasts are actually moving, whoopsie, they're moving around inside of the cytoplasm. Let's check it out. What happens here is that these actin microfilaments are grabbing onto the organelles, like the chloroplasts, and sort of pulling them along. And they're handing them off from one filament to another, causing this movement of the cytoplasm and then pulling the chloroplasts in that direction. So they're all moving around in there, and that has certain advantages. If you think about it, that's actually a good thing because it distributes nutrients and energy and things like that, ATP and all of that business, all around the cell, allowing all parts of the cell to get what it needs. Also, if like the sunlight is over here on this part of the plant and not over here, these chloroplasts sort of all get a chance at the light by being able to move around all over the cell like this. So we call that cytoplasmic streaming. Let's check it out. I think I have a video here. Let's see. Yes, I do. So this would be Elodea cells, which is a water kind of plant, and the chloroplasts in here. This is magnified pretty highly, but the chloroplasts are moving around inside of the cytoplasm due to the action of these microfilaments. You cannot see the microfilaments on this. They're much too small to see. All you're seeing is a little bit of movement in the cytoplasm and then the movement of the chloroplasts. 1250 light microscope magnification. My goodness, that's at the limits of light microscopes. Very high quality microscope, clearly, because of the quality of this video. Beautiful. All right, gotta love it. All right, so that's cytoplasmic streaming. Another function, although a little bit obscure because it's not in most cells, both of these, only in some cells, not every cell. Although actin is present in almost every cell. All right, I want to make sure I covered everything about microfilaments. Cytoplasmic streaming we talked about, muscle contraction we talked about, changes in cell shape and cell motility kind of go together. Oh, cell division. See, I got to insert another slide. My goodness, unprepared. Okay, so cell division we need to add. Let's drop that in here. Role of microtubules in cytokinesis. All right, so cytokinesis is when the cell splits right at the end of telophase in mitosis. Let's check it out. right at the end of mitosis, when the nuclei are starting to form, what you're going to see happening with cells is there's this pinching of the membrane towards the middle. This gets pinched that way, this gets pinched this way, and the nuclei are forming again over here. Everything is coming back together. They're about to split. So there's microfilaments inside here that grab onto the membrane and pull it. They're pulling it down over here, they're pulling it up over here, until eventually you're going to form two separate cells. Okay, so that cytokinesis, or pinching of the membrane, the splitting of the cells, we call that cleavage sometimes too, is thanks to microtubules, thanks to actin filaments. Actin and microtubules, I'm sorry, oh my goodness, I wrote microtubules. I hope you caught that. Microfilaments. Good thing I caught that before I finished the video, my goodness. I'm not gonna like edit this out and re-record it. I'm just gonna leave it in. You're gonna have to deal with it. All right, now there's no evidence of it anymore. Microfilaments and actin, basically interchangeable. All right, I think that covers everything from microfilaments. Let's move on to those intermediate filaments. These are pretty boring. They don't do anything super exciting. They're medium. They're like the middle sister. They're basically Jan from the Brady Bunch. They're medium in size. You don't have to worry about the diameter, nanometers, whatever. Forget about that. They're made out of keratin. Okay, so that's good to know. That's the same stuff like hair and fingernails is made out of. They support the cells, so they maintain cell shape just like the other guys do. The other two things that they do is they form the nuclear lamina. So inside of the nucleus, if you look at the inside wall, there's this sort of lattice of fibers. Those are intermediate filaments, and they strengthen the nucleus. They prevent it from getting smushed. The other thing that they do is inside of the cell, they will anchor the nucleus. So if this is the nucleus, it's held in place by these intermediate filaments. And they're sort of kind of like if you have an antenna. If you have an antenna pole like this, you know, there's these cables that hold it in place, and the cables are kind of stretched in all the directions to hold it in place so it can't fall this way or that way. And that's the same thing here. This prevents the nucleus, when the cell gets like distorted, from going all the way to one side of the cell. And now the nucleus is over here and it's kind of stuck here. These intermediate filaments prevent that from happening. They keep it, you know, pretty much in the center of the cell where it belongs. So anchoring organelles, especially the nucleus, nuclear lamina, and then maintaining cell shape is intermediate filaments. Overall, not super exciting, nothing super high-tech, more structural support kind of stuff. Nothing fancy like cell division or movement or anything like that. All right, so that's intermediate filaments, and that is the end of our cytoskeleton discussion. There was a lot of stuff I talked about with the cytoskeleton, a lot of stuff to learn. So make sure you study this well, write it out, like take blank sheets of paper and write out, you know, microtubules. What do they all do? Write out, you know, do little bullets and figure it all out. Microfilaments, there's lots for both of those, less so for the intermediate. So, you know, if I'm going to ask you about one, I'll probably ask you about the first two. You know, if it's like a free response or something. Maybe I'll do a multiple choice on the intermediate because they're not that exciting. All right, so we know that plant cells, let's change gears a little bit. We know that plant cells have a cell wall on the outside that makes them really strong. And that gives them their shape, that squarish kind of shape that plant cells have, versus animal cells being more sort of amorphous, like lots of different shapes. So cell walls strengthens the plant cell, you know, and keeps its shape, blah, blah, blah. Do animal cells have anything like that? Well, they don't have a cell wall, but what they have is the extracellular matrix. And that sort of takes the place of the cell wall in an animal cell to help maintain its shape and give it strength, because the cell membrane is really flimsy and really thin and not really very strong. But the extracellular matrix has other functions too. It has like communication functions where cells can talk to each other and send signals to each other via these kind of fibers here, these proteoglycan fibers. They can act like antennas. They can send these signals down into the cell through the integrin, through this fibronectin thing. and then the cytoskeleton can pick up the signal and send a message somewhere into the cell, which is kind of cool. Integrin is kind of easy to remember because it integrates the membrane. It's an integral protein all the way from the outside to the inside. It passes all the way through. Fibronectin is not really all that important. Proteoglycan, I don't really care too much if you memorize that. However, if I asked you proteoglycan, if I gave you that term on a test and I said, what do you think it means? I would hope you'd come up with protein over here on the left and sugar over here. So glyco or glyca is sugar, and then proteo is protein. So they're made out of protein and carbohydrates, sort of a hybrid kind of a molecule. All right, that's the extracellular matrix. Let's talk about connections between cells. In plant cells, sometimes there's like tunnels, like passages between the cells. They're called plasmodesmata. And they allow materials and things, hormones and other kind of molecules, to travel from one cell to the other. It's little tubes. You can see them on this electron micrograph. There's actually like a little tube connection between the plant cells that kind of goes through the cell walls like that. That's kind of neat. All right. Do animal cells have anything like that? Well, let's find out. Animal cells actually have three kinds of connections between them. They're tight junctions, desmosomes, and gap junctions. Tight junctions basically act like caulk. So they seal the two cells to prevent water from passing between the cells. And this is important in places like the small intestine. So what happens is that you don't want materials from the inside of the small intestine to sort of pass through and get to the bloodstream in between the cells. What happens is molecules like small polypeptides, amino acids, triglycerides, things like that, pass through these cells of the small intestines before they get to the bloodstream, and they get processed inside of the cells and broken down a little bit further into, let's say, fatty acids and glycerol, stuff like that. So it's important that if you have cells like this in the wall of the intestines, that the water and the material from the lumen of the intestines does not pass in between. That's not what you want. You want it to go into the cell, pass through here, get processed, and then enter the bloodstream over here. All right, so that's a tight junction, kind of like caulk. I'll show you a picture in a second. Desmosomes are just like rivets that kind of anchor them together and strengthen the connection between the cells. So not super exciting, but important. And then finally, gap junctions are little channels between the cells where materials can move between. Those are kind of like what in plant cells? Did you say plasmodesmata? You are correct. So in animals, they're called gap junctions. In plants, they're called plasmodesmata. They're equivalent. Different structure or whatever, but similar function. Don't confuse desmata with desmosomes. That's not the same thing. Different function. All right, so here this picture shows the tight junction, which is like the caulk that seals them. You might not have all three of these all in the same cell like this. That's sort of just a fictional example. Desmosomes to anchor them between the two cells, like a rivet on like a pair of jeans. And then gap junctions are little tunnels, just like the plasmodesmata in plant cells. All right, and here it shows, basically this shows intestinal cells, and you don't want water to get all the way through here. You don't want that going into the bloodstream in between the cells. You want the water and the nutrients, the molecules, to pass through the actual cell. So you seal that with the tight junctions, like caulk in a shower stall or in a window to prevent water from getting through. This is just a quick review of all the parts of the cell that we talked about. You can look through that on your own copy of this PowerPoint, and then you can answer these questions because they are a nice little review of Chapter 6. All right, thanks for listening.